mostly talk about work from my postdoc, but I will try to get also to a little bit of newer stuff towards the end. And indeed, I started with electrical engineering and made the decision that I don't want to make money and move to science. And I did my PhD at the Weizmann Institute, working on the whisker system of rodents. And then I decided that I want to move to a model system that enables high resolution circuit analysis. And this was the motivation to move to Drosophila. I studied social behavior in Malamurthy's lab at Princeton. And then in my own lab, I'm also now focusing on the neural basis of social communication and working on Drosophila as a model system. So I do want to spend a couple of minutes to emphasize what, at least for the type of questions I'm asking, why Drosophila is a good model system. And I think it boils down to 2 points. One of them is complexity, the other one is tools. In terms of complexity, the fly is, on on logarithmic scale, is like halfway between worms and rodents. And this means that it has complex behaviors that are, that we can quantify and they are rich enough, mating behavior, aggressive behaviors, for example. But at the same time, we are able to dissect circuits to the level of very few neurons, as I will show you, even to the level of single neurons sometimes. So the complexity is really key. Here, the other thing is the tools, and given the history of Drosophila as a model system for inheritance and for development, we have very strong and powerful tools that enable us to focus on very specific circuits for monitoring them or perturbing them. And more recently, we now have the complete connectome of the fly, which is really a great thing. The brain of the fly has around 127,000 neurons, and we have each one of them tracked. And we have all the synaptic connectivity between them, even some predictions about the synapse types. So with this, we really have a big map that helps us to, move in a kind of consistent way to understand circuits. And we go back and forth from finding new cells and circuits in Flywire, building genetic drivers, and so forth. Now, with new tools for quantifying behaviors, mating behavior, for example, is happening in Drosophila melanogaster in 2 dimensions, so we can track the flies at very high resolution with various tools that exist today, which automate the behavioral quantification. This is, of course, very important. And lastly, together with the size of the fly, we can also image throughout the fly brain with a 2-photon microscope, right? So all the way top to down in the fly, that's another big advantage. Okay. Now, the question that I really want to understand is what's the neural basis of social communication? And when think about these people that drink coffee in this coffee shop, then they have to make decisions all the time about their reaction, what they're going to say, what they're going to do. And these decisions are based on a few things. They are based on sensory integration of multiple cues. Many times, in the case of social communication, multisensory, so for example, sounds from other people, maybe even the smell of the coffee in the air. And it also depends on the internal states of the people. Are they angry? Are they afraid? Are they happy? Are they not happy? This internal states reflects many things, including the context and the needs of the individuals. And of course, it also depends on the experience. So all of these things are not unique only to social behavior, but they are very strong in the context of social behavior. In the case of fruit flies, here you can see a male fly courting a female fly. So he is at the back, and you see he's running, and once in a while you see he's moving one wing, he's vibrating one wing. This is his way to sing to the female. This ritual takes typically a few minutes until the female makes a decision, and this decision could be to say yes to the male, and then they will copulate, or to say no to the male, then she can jump away, and the male can also move to another female. So this ritual is actually quite multisensory. You can see now the auditory and visual part, but it also involves pheromones, and also it involves touching. And interestingly, the choice of the male to sing one song or another depends, that was once thought to be random, is actually, you could predict it by visual feedback that comes from the female, and also by the motion of the male itself. So there is some interaction between them. He's changing his song in reaction to the female. The female's decision to mate or not to mate, but also to slow down or accelerate, depends both on on the singing of the male, but also on her own state. So for example, a mated female behaves completely different compared to a receptive female, right? So the state of the female, the internal state of the female, has a big effect on the way she's going to react to exactly the same sensory stimulus that comes from the male. However, even though most of the experiments that we and others do with Drosophila, we put one male and one female, in nature, it looks more like this, like it looks maybe in your kitchen sometimes, that you have many flies together. This is where they, or hopefully not, but maybe this is where they mate, this is where they eat, this is how they live. And so it looks very, very different, and I hope to get back to this towards the end. But what I want to say now is that even though in the case of Drosophila melanicus, only males are singing, and they are singing to the female, as in the movie I showed you, the song is actually very important also to the males. Other males hear the song, other males around. And we know, for example, that if we introduce with a speaker, just playback of a courtship song, in this example, the pulse song, then the males are starting this chasing behavior. This is what happens in the lab. And another thing we know from a more recent study is that if you put two males to compete over one female, then one male sometimes is making some noises that are actually jamming or overriding the song of the other male. So they hear one male singing, and as a competition, they try to interfere. So these are two examples to show that the males also care about the singing of other males. Now, I want to tell you about two stories that came from my postdoc. The first one will be focused on the sensory processing part of the social communication, and I will touch a little bit on experience, but not much. And I will ask what are the shared and sexually dimorphic circuits for processing the courtship song. The second story will be focused on the internal state, and specifically internal states in the female brain and how they affect her response to signals that come from the male. So starting from the first question specifically is how do similar auditory cues, the song of the male, drive sex-dependent responses. And in a somewhat simplified way, you can think of two models. One of them is that there are completely parallel or different circuits all the way from the receptors to the behavior in males and females, such that they drive different behaviors, or it's also possible that some part of the circuit is shared between males and females, but somewhere it diverges such that eventually males and females show different response to the same sensory signal, to the same auditory signal. Now, this is the courtship song of the flies. Let's see how the auditory thing works here. But you can see that it has two major components. One of them is the pulse song. So these are like short pulses that come out one after the other. The other one is sign song. And you can see here that in the single bout of singing, there is often a mixture of these two components. Now let's see what sound is. Through the room microphone speakers, but so maybe in the back you couldn't hear, but the pulses are like tick 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 thing, and the sign tone is just humming. It's a pure tone that sounds like something like this. And as I said before, the change or the moving from one type to the other depends on visual feedback from the female. So it's very homogeneous. Now we wanted to ask how males and females respond to this courtship song, and we decided to do it with this playback essay where we have a speaker and we introduce courtship song, but we could also introduce different components of the courtship song. The reason we don't do it during natural behavior is both because what I told you, that there is this confound that when the male is singing, the female reacts to the male and the male reacts to the female, so it's hard to separate, but also because we wanted to be able to span a large number of parameters in a systematic way. So we took in some experiments only pulse song with a specific interpulse interval, or sign song with a specific frequency, etc. So we played systematically with many parameters of the courtship song, eventually trying more than 50 stimuli to see, and we checked everything separately in males and females, right? So you have individual flies, each one in a separate track, and then the thing that we measured here mostly is the locomotion. One more thing that I'll talk about later. So here you see that courtship song has multiple time scales that describes it. One of them is this, the bout duration that could be a second or even several seconds sometimes. And then the interpulse interval is in the order of tens of milliseconds. And then the carrier frequency of either the pulse song or the sign song they are in the time scale of milliseconds, right? So this is how the song looked like, and we played with each one of the parameters. I want to mention here that some of the parameters, the faster one, like the interpulse interval, for example, is different between species. So Drosophila melanogaster and Drosophila simulans, for example, they have different interpulse intervals. So one of the type of information that the song carries is about the species. The other, when we look at longer time scales, then you see that it's not different between species, but it is different between individual males depending on the interaction with the female, and males maybe also differences between males. So these are the different time scales. So here is how the experiment looks like in a single, when this is an example for a single stimulus. So here we have 4 seconds of train with pulse song with 36 milliseconds IPI, for example, and we look at the change in this example of the female speed, right? At some window, and then in one experiment we try different interpulse intervals in a pseudo-random way, and eventually we can measure what is the response to different interpulse intervals, right? So we get this tuning curve, this behavioral tuning curve, and you can see that the female is slowing down in response to pulse song, and you can also see that she slows down mostly when the interpulse interval is around 36 milliseconds. And this is consistent with the fact that Drosophila melanogaster sings pulse song with an interpulse interval around 36 milliseconds. So she's really tuned to her own species. Now, this is with the interpulse interval, and we do the same thing with the male, and you can see something that's interesting, which is that the male is actually accelerating in response to pulse song. However, yes?
Does it matter if it's his song or someone or a different, or one that you created?
So we did it only with a speaker from the outside. The question whether the male hears its own song in response to your own song is the short answer is that we don't know exactly, but I can say more about this. But in this case, it comes from the outside. But what I also wanted to emphasize is that the peak is exactly the same place. So they are both tuned to Drosophila melanogaster song, but the females slow down, the males accelerate. So this is sexually dimorphic response to pulse song, and this is true not also for IPI. They are tuned for other aspects of the courtship song, but in all cases, with pulse song, the females slow down and the males accelerate. So this is a sexually dimorphic response to pulse song. In the case of sine song, the response is much less different between the males and the females, but specifically the response to pulse song is strongly sexually dimorphic. The other thing we found is that on top of the males accelerating in response to hearing the song, it also drives singing in the males. So they hear pulse song and it drives pulse song in the males. Why? We can speculate. If a male is singing, it means that there is another female around, etc. But the fact is that this is what happens, and here again, if he hears the right song, then it drives singing and more singing. And it's not happening in the female. The females never sing. There are artificial ways to make them sing, but they never sing in response to courtship song or just naturally. So looking at these results, because the tuning is the same between the males and the females, but it drives different behavior, what we think is that this model is more appropriate. It looks like the sensory processing, right? The tuning is the same between males and the females. They are both tuned to the specific frequency and to specific interpulse interval and specific pause between the pulses, duration of each pulse, but it eventually drives different behaviors in males and females. So this is just from the behavior, and now our next step is to look for this feature detector in the brain of males and females and see if indeed we can find a place where the tuning is the same between males and females, but when you activate it, it drives sexually dimorphic behavior, right? So this is bringing us to the part where we look into the brain. So we put the flies, males or females, under the 2-photon microscope, and we introduce the same list of sounds with a small speaker, and then we measure calcium response. I will assume that we use GCAMP, and I assume that people here know, but generally it's a way to look at neural activity in the brain. And so we focus on this area. So this is the arista, this is the hearing organ of the flies, and we look at this area where there are third order neurons for auditory processing. And the reason that we focus on this area is mostly because we know that this area is very rich in neurons that express these two genes. Now, double sex and fruitless are two genes that control most of the sexual dimorphism in Drosophila, both morphologically and behaviorally. And people already know that neurons that express one of these two genes, or both of them, are often controlling sexual behaviors. So we focused on this area that's very rich in neurons that express these genes, and specifically we focused on double sex based on some previous literature. So these are the double sex expressing neurons. In this case, I'm showing it in the female brain. They have names. So this is one group, and this is a separate group, etc. And we imaged them under 2-photon microscope. And what we found is that there are few groups within the double sex expressing neurons in this area that responds to pulse song and have very weak response to sine song. And all of them are tuned to all these different parameters of the pulse song that also the behaviors is tuned to, right? And the largest groups is the PC2L neurons, right? And the response or the tuning of the males and the females in these neurons is similar. And you can see it here. So if each dot here is a different stimulus, right? Sine song with 100 hertz, for example, or pulse song with a specific pulse interval, and you can see what's the response in the male and what's the response in the female, right? The delta F over F, the calcium response. And you can see that in the case of a sine song, the response is very weak in males and in females. In the case of pulse song, there is a strong linear correlation between the two, right? So if the female responds more to the stimulus, the male also responds more to the stimulus. So this is in this area, and here we also correlate it with the behavior. So in the case of the female, the male, the sine song again has very low neural response. But if we look at the neural response and we look at the behavioral response, there is negative correlation in females and positive correlation with males because females slow down and males accelerate in response to the stimuli. Yeah,
right. What is each point on the plot?
What is what?
What is each point on the plot?
Each point?
Yeah. What is each circle?
No, sorry. Each point is one stimulus. That's what I said. So for example, one point could be when you introduce sine song with 200 hertz frequency. That's one point. And then we can see what's the behavioral response. And we can see what's the neural response.
And how many flies is that?
It's over, how many flies is averaged over. Yeah. In the case of the behavior, it's averaged over 120 flies. In the case of the brain, it's averaged over something around 10 flies.
Right. So each point should have some sort of variance on it, right? Because you have variability. So how big is that variability?
Okay, good question. The variability in the neural response is not large. They are always tuned to exactly the same, or within the resolution we check to the same frequency, etc. There is definitely a lot of variability in the behavior for multiple reasons, including the fact that when you introduce a sound, some flies are not moving at all, or are very fast, so their potential to accelerate. But in the behavior, there is definitely variability. This is why we average over so many flies.
Well, but also the neural response, right? So how many right? There's variability in the neurons, there's variability over the individuals, right? And it's like this is like individual neural averaging over both of those.
So this is again, each point is averaged over, in the case of the neural activity, averaged over 10 flies. So for each fly, and also for each fly over a few repetitions, not many, because in one experiment we try many stimuli.
But how many neurons? Right, it's delta F over F.
So this depends a little bit. In the case of the PC2L, there are about 8 neurons that respond. And these are little details depending on, in some cases we look at individual neurons, in other cases we look at some area that integrates a few neurons, but this is the number of neurons that have this response.
Can you measure suppressive responses in the calcium?
Can we measure what response?
Suppressive responses.
So if this it doesn't happen here, but generally yes.
The question is, if you have a suppression then rebound, would you not confound it with positive delta F?
So, but it's not happening. So we do have other experiments, not here, where not my work, but from the same lab, where we looked at auditory response throughout the brain, and we can definitely see also, some inhibitory areas, etc. It looks different. We don't have, at least not the resolution that we can measure with calcium. We don't see.
So I wonder whether the conclusion that there are all similar responses may be that actually suppressive responses in the female, in the normal responses, which has, you have suppression, you have rebound, and you count it as delta S, positive delta A.
Okay, so this is defined as the average over the window that starts at the onset of the stimulus and ends a second after the offset, and this is what we measure. We don't have the higher resolution dynamics. So yes. Okay, so from this we conclude that we see here that there is this group of neurons, this PC2L neurons, and some other neurons that have similar tunings between females and males. And now the question is, what happens if we activate them, right? So if we activate them, can we now get this sexually dimorphic behavior? So now we use channel rhodopsin to do it. It's redshifted, but not important. And we created this intersection so we can look specifically at the PC2L neurons, this subgroup. And when we activate these neurons, so the first thing that you can see here is about this singing phenotype. If we activate the PC2L neurons in the males, it drives singing. It drives singing of pulse song, and there is another phenomenon that when the pulse song when the stimulus goes down, pulse song goes down, and then there is a real sign song. There is more to say about this, but the point here is that we definitely can create the males very robustly sing pulse song by activating the PC2L neurons. So they're really at the interface, the sensory motor interface. They are tuned to pulse song, but they also drive pulse song. When we do it with the female, we never get singing. So that's definitely this phenotype. In the case of locomotion, we also get sexually dimorphic responses, very, very different between males and females. I will say that it doesn't look exactly like at the behavior. This could be for multiple reasons, but I will say that we don't really think that PC2L neurons are the only pathway from the auditory side to the locomotion. It's more complex than this. But we definitely do see sexually dimorphic response in locomotion when we activate these PC2L neurons. Okay, so this was this part, and the summary is that this is the model that we think is more correct, and PC2L neurons seems to be this node, and maybe there are more nodes. And I will mention very briefly here that we also looked at the effect of experience. Flies, the males don't have to learn to sing, the females don't have to learn to be tuned to a specific song, but it does turn out that when we group house the flies, then in the case of males, but not in the case of females, the tuning becomes narrower, sharper, around the 36 millisecond interval, and another group followed up, and they also showed that you can do it with a playback. So when you raise the males or the females when they hear the pulse song for some time, then it sharpens their tuning. And we also found that in these PC2L neurons that are in the sensory motor interface, we can see this learning already. So this is just to say that there is also learning in this system.
Over what duration? Like, what's the experience like?
So here in this case, we just raise them from their eclosion until they are around 4 days old. There is some window all the time, in a group or single house. But they did experiments where they also did it just a few hours a day, still for a few days. I don't know what's the meaning. Okay, so this is concluding the first story. And now I want to talk about internal states. That's the second question. And I will start by mentioning this review from David Alderson that talked about some similarity between activating specific neurons in the hypothalamus of mice that drives both mating and aggressive behaviors, specific attack and mounting, and the activation of a small subset of neurons in the male fly that also drives both singing and aggression. And this is the experiment, or one experiment that shows it. If you activate this small subset, this P1 neuron in the male, what you can see is that there is first singing during the time of this activation, and then if there is another male that's in the environment, then afterwards the singing goes down and aggression goes up. What's important to see here also is the persistency of the effect, meaning that the activation happens here within this 1 minute time window, but then the aggression persists for many, many minutes, more than 10 minutes after the end of the activation, right? So don't activate anymore. And there are a few things that characterize internal states, and I think 2 of them are the persistency of the internal state, the fact that they last for some time, and also their scalability. So I'm not showing it here, but when they try different stronger and weaker stimuli, then you get more or less of the effect, and also more or less of this persistency. So they showed that there is some group of neurons in the male that you activate and drives this persistent behavior. We wanted to look at the female and see if we can get some similar phenotypes, taking into advantage, as I'll show you later, also the connectome that we also only have in the female so far to go further towards the circuit. So these P1 neurons that I mentioned that drives the thing in the male, this is actually a small group that exists only in males, male flies, but they are part of a larger group that also exists in the female. So we wanted to ask if these neurons that also exist in the female also drive some persistent internal state in the female. The PC1 neurons, we'll talk about them now. We talked about the PC2L neurons in the previous story. Both of them express this double sex gene. So we are going to measure how, if there is a persistent effect of activating PC1 on the female receptivity, that's one thing. If it affects persistently a response to the song of the male, and it also the moment-by-moment social interaction. So this is how the experiment looks like. We put a female, we activate the PC1 neurons. In the first experiment, we did it for 5 minutes, then we tried 1 minute and even half a minute, but this is the 5 minutes. The female is there alone. And then we add the male, and then we start measuring things, right? And then in other variants of these experiments, we do the same thing, activate the female alone for 5 minutes, but we wait for either 3 or even 6 minutes before we add the male, with the idea that if it drives some persistent activity in the female brain that lasts for, and we can still see a phenotype here, it means that something lasted in the female brain for these 3 minutes, or maybe for these 6 minutes even, right? That's the idea, to measure the persistency by looking at the behavior. So the first thing we measure is receptivity, and you can see that here is showing the percent of lies that are copulated after 5 minutes, and you can see that you get a big effect on the no delay condition. You still have an effect, and those pictures are still significant on 3-minute delay condition, but when you wait for 6 minutes, there is no effect anymore, okay? So we have a persistent effect on the female receptivity. They copulate faster with the male, much faster. So the second thing we looked at is the response of the female to the courtship song of the male. I'm showing you here how the setup looks like. So we have these tiny little microphones to record the singing of the male, and we also have a camera that allows us to track the males and the females. And when we do this, we look at the response of the female to the song of the male. The negative correlation here means that the females are slowing down. This is on time scales of minutes. And you see a big effect only in the no delay condition, right? So only when immediately after we stop activating in the female, we add the male, we have a strong effect on the female response. But if we wait for 3 minutes, this effect is gone. And then the last thing is to really look into the details of the behavior. So for this, we used LEAP. Now there is another version of it, but essentially you train some neural network to find specific points on the fly, and then from this we extract a few features like the male-female distance, the angle between them, etc. 17 parameters, and I will skip a little bit the details here, but we did use some unsupervised approach, and eventually what we found. So the first thing you can see here is that this is what happens when we train a classifier to compare frames with the features that come from controls versus all the other conditions. And then we try to see how good this classifier works on the different specific conditions, on the 0 minute or 3 or 6. You can see that the strongest effect is actually on the 3-minute delay. So the behavior of the female on the 3-minute delay condition is very, very different from the control, and also compared to the 0 minutes. So this is a bit surprising. Maybe it will be more clear in the next slide. But the point is that we have a stronger effect when we have the 3-minute delay. And some more details that maybe I will skip about the clustering, etc. But eventually we found these two specific behaviors that this is unsupervised, so we just call them behavior 1 and 2. But when we look at the movies, we can see that these are the behaviors. I'll show you a movie in a second, but one of them is the female shoving, the other one is the female chasing, and you can see here again, this is how much these behaviors happen in the control, this is how much they happen when we have a no delay condition, And you can see that both of these behaviors happen much more in the 3-minute delay condition. That's maybe almost surprising, because when we wait for 3 minutes and then we add the male, we get much more shoving and chasing. And this is how it looks like. So here you have the male has a dot on its back. This is the shoving behavior. Face to face, the female is pushing the male back. This is an aggressive behavior, and this is chasing. The female is chasing the male. This is usually not happening in flies. So the female is chasing the male for some time. They do it a lot. Very rare when we just put naive males and females. And eventually they copulate. Not always, of course, this is happy end, but actually it happens quite a lot that there is this aggressive behavior. So the female will shove the male, and shove the male, and shove the male, and then they copulate. So that's a summary of this. So there is an effect on the response, the locomotion response that peaks at the no delay condition, receptivity I'm not showing here, and there is also this modulation in the behavior, this aggressive behavior that actually peaks at the 3-minute delay condition. So there are two time scales. The peaks of these two phenomena happen at different times.
This is across the entire 30 minutes?
This is across the entire 30 minutes. Or until they copulate. So if they copulate, we stop measure. Right?
Any difference over time? So in the first 3
Yeah, it's a good question. I actually don't have a slide for this, but yes, there is some dynamics. So for the 3-minute, it peaks after. I had a slide, I removed it, but it peaks after 2-3 minutes after we introduce the male, and then it goes down slowly, not even to zero, but it goes down until the end of the 3-minute window. And many times the flies they copulate after 5 minutes or so, and then we stop measuring because they copulated. So now because we found that driving activity in PC1 neurons drives different phenotypes, and specifically receptivity and aggression, then we asked ourselves what is actually happening. And you can think of a few options. One of them is that there is, PC1 is a homogeneous population that downstream, drives different populations. One of them drives chasing, the other drives shoving, the other one drives receptivity. An alternative is that we actually are activating a heterogeneous population, and different subsets of the PC1 neurons drive, these different phenotypes that we find. But eventually what we found is this, that there is one subset of the PC1 neurons that drives both the shoving and the chasing, but there is another group within the PC1. And how do we get to this conclusion? I will say now. So this is where the connectome of the fly is very important. We have 2 connectomes now of the female fly. They are not exactly the same, but generally 2 connectomes. And this is really great because we could find the PC1 neurons in this connectome. And when we did it, we found that PC1 actually has 5 subtypes: PC1A, PC1B, PC1E. And we use the driver of PC1B and PC1E, so a driver that is just for these 2 subtypes. You can see that there are similarities between them, but they don't look exactly the same. And when we use only this driver for PC1, B, and E, then we get the aggressive phenotype, but we don't have any effect on receptivity. And now, the other thing that happens is that now we don't have anymore this effect that there is like a much stronger response in the 3-minute delay condition. And so with more experiments, and I'm not showing all of them, okay, actually, sorry, I wanted to say another thing. So the PC1 D and E neurons, this subset, is enough to drive the shoving and chasing. This is how we concluded and got to this conclusion. And looking at the connectome, we also found all the connections of these neurons with other neurons. And one thing that was very clear, that there is another group of sexually dimorphic neurons that is very strongly reciprocally connected, right? So they are connected in both sides with this PC1 D and E neurons. These neurons are called AIPG. Right, so you can see here maybe you can see the morphology of the neurons in this movie. These are the PC1 neurons, and then you can see all of these different types of AIPG neurons. They are all reciprocally connected with PC1 neurons. So we can't say for sure, but this drove us to think that maybe this reciprocal connectivity between this very small group of PC1 D and E neurons and a larger group of AIPG neurons, maybe this recurrent connectivity could be part of the explanation for this long persistent activity, even though we can't say it just by looking at the connectome. So if this is true, we would expect to see persistent activity in both the PC1 neurons and also this AIPG neuron. So here we looked into the brain, and we ran the same protocol, only that now we activated in the brain for 5 minutes and looked throughout the brain. So this is pan-neuronal two-photon imaging. And what we found is that in some area of the brain, we get response during the activation only. So this is not persistent activity, but we also found some regions of interest on some areas in the brain where we can find this type of persistent activity. So we see activity that outlasts the stimulus for more than 5 minutes. So now we know where the persistent activity is in the brain, even though this is still not at the resolution of single cells, because when we do whole brain imaging, we can't point on specific cells. So now more work will be needed to find specific cells, but we did a little bit more of this by looking at some specific population, double-sex neurons and the fruitless, and we could find persistent activity also in individual neurons. So we know where this persistent activity is, and this model, I'm not showing you all the data for why we are thinking, but this explains this model with some other, behavioral results that we have. We go to the conclusion that we think that this group of neurons that drives the receptivity is actually inhibiting some of the neurons that drive mostly the shoving behavior, and this could explain this peak at the 3 minutes, because the receptivity is peaked in the first minutes and it drives down the aggression. But once the persistency that drives the receptivity disappears, then the aggression goes up. I can say more about this if someone is interested. Now, I want to say that similar work was and still being done in rodents, including a lot from David Anderson's lab. This is an example in mouse fear, and in this case, they are not activating artificially with optogenetics, but they're actually showing the mouse a rat or a toy, and they can get this persistent activity in specific neurons in the hypothalamus. You want to ask something?
Yeah, just you're running very fast. The PC1DE are sexually dimorphic.
So all the PC1 neurons are sexually dimorphic, and specifically also the PC1D neurons. Sexually dimorphic could be many things, including in one extreme the P1 neurons that only exist in males, I said shortly before. These neurons, the morphology is different between males and females. It's also a little bit about terminology. There are PC1s in males and females, and they look differently. So if they define them exactly by morphology, then they don't exist in the male. But there are somewhat similar ones in males. I also have to say that male connectome is only now being almost complete, so we don't have the full risk. We don't have all the neurons, individual neurons in the males yet.
But you did the optogenetic experiments in the males as well?
No, they did it, so we were motivated by other work that was done in this P1, the subset of PC1, so this was already done before us, and they found this persistent singing and persistent aggression in males. So this was before us, but we did it in females. Yes?
Do you see this persistent activity in natural contexts where there's no optogenetics? Does that happen on the one?
Right. So this is in mice, and they do it without optogenetics, right? But if you ask if we already have the condition to drive this persistent activity just by showing an outside stimulus, the answer is no. And I think it's an important thing to do, but we don't.
I want to follow up on Aya's question before. So, in the delay experiments, essentially in the no delay versus the 3 minute delay, in the no delay, after the first 3 minutes have passed, are the behaviors similar?
No, they are not. So you mean if you compare, just to understand you correctly, so if you compare the no delay, but you look 3 minutes after, versus you look at the 3 minute delay and you look at exactly at the moment of the beginning, so it's the same time, very different. Even more extreme is the 6 minute delay. In the 6 minute delay, you barely see any aggression. But if you do the 3 minute delay, the aggression lasts much more than 3 minutes. So I think what happens is that there is still something in the brain, but then now you add the male, and this, adds another stimulus, so now it lasts for a lot of time. But without the male, it decays after less than 6 minutes, somewhere between 3 to 6, almost to 0. There is also variability between flies, so sometimes they show it even after 6 minutes for some flies, but not many. And I would just say here that this is part of my future work here to try and understand what are the mechanisms. So in this work, they suggested this model where there is a combination between neuromodulation that allows this long time scale and between local recurrent connectivity that allows specificity. So this is their work in mice, but this is a challenge now to understand really where the persistent activity is. Does it really depend on recurrent connectivity? are neuromodulators already involved? Which neuromodulators, etc.? These are open questions that for me are very interesting. So now, tell me if I have a few more minutes, or I do. Oh. So that's great. So I want to spend a few minutes talking about future directions with just a few results, but let's do it. So, one direction now is to move from 2 flies to many flies, and I talked about this a little bit at the beginning. And so now we track all these individual flies and look at the dynamics. One thing that's very, maybe not surprising, but very clear when you look at the dynamics of multiple males and multiple females together, is that unlike in the experiment when we only have one male and the male would chase the female forever if they don't copulate. So this is the distribution here. We stop the experiments after 30 minutes, so it can't be more than this. But if we ask how much the male would stay persistently and court the female without leaving her alone, then this can last for many, many minutes. You just keep trying and keep trying, they don't give up. But if we put all these males together with in this case 4 males and 4 females, what actually happens if you look is that the males, they court a female for some time, then they move to another female, then they move to another female. All the dynamics is completely different, and at least at this small pilot experiment, we never saw a male that just look at one female for many, many minutes. Other males come, they compete. One male, another male come, one male gives up, etc., etc. So the dynamics is completely different when you have multiple flies. And what we try to do now is so what we here show there is this egocentric views. We try to look at the world from the point of view of each individual male or female and try to understand or to predict what the decisions are going to be. So for example, and there are also models for this, but what would be the right strategy and what is actually the strategy of the male deciding whether you want to consistently or persistently court one female versus just move to the next female, right? So I think it's an interesting question in general, but also in flies. And this is one thing that we are trying to understand now in the lab. And another direction is to now add other modalities. So we talked about auditory stimulus only, but we know, as I told you, that other things are involved. We know that pheromones, for example, are very important. And when we looked at this, experiments at the PC1 and PC2 neurons, these double-sex expressing neurons, we found and we tried so far just 3 pheromones that we thought might be relevant based on the literature. Then we found clear responses in PC1 and PC2 neurons to different pheromones. What's interesting and still I don't know why exactly is that we found both excitatory and inhibitory responses. In some cases it's expected based on the ethology. In some cases I think it's less clear. What we still didn't do, and that's the next step, is to try in the same cells to look at the auditory response and the pheromone response and put them together and see. Because I didn't say it, but just the same way as there are different PC1 neurons, there are also different PC2 neurons. And so it's possible that different population, even though they all have the same response. But when you add pheromones, maybe now you will start to see differences. Maybe some neurons are integrating pheromones and sound, and some neurons are integrating sound with vision or whatever. We don't know this, but we try to understand. One of our hypotheses that we want to try to test is whether the existence of pheromones change the integration window. So this very slow signal of pheromone, does it change the way that the female or male integrate over the fast auditory signal? And this is just one idea that we want to test, right? So actually, I didn't say it, but this is the little tube that we use. This is also a screen, but we will get to the visual thing later. So eventually, the idea is to have this multi-sensory environment that has everything, but we do it step by step. The other direction is to kind of follow up on the work on the sexual dimorphism and understand where is the sexual dimorphism downstream these PC2L neurons. So we can image also from this nice fly that walks on the ball, in which case we can measure both the locomotional behavior and the neural activity, and we can optogenetically activate different neurons and try to understand what's different in the males and the females. I will also mention here in the word, in one word, that the females, I told you that they slow down and the males accelerate, but the fact is that mated females accelerate. So it's not only differences between males and females, also that females in some state behave like males. So is it the case that the same circuit allowed both options, but the weight somehow are different between males and females, and also weight different between virgin and mated females? Is it the same circuitry that allows both types of flexibility or not. The hypothesis that I have is that the answer is yes, but I don't know. Say one more direction. And this is follow up on the work on the persistent activity, and what we do here is we try to think where this persistent activity could be. The effort here is to use the full connectome of the Drosophila brain to predict where we think this persistent activity is. So we looked at different types of neurons. This is a new work in preparation where we found all the double sex and fruitless neurons in this connectome, or at least based on morphology. We can't really measure the double sex and fruitless expression. And so based on this, we found all these different types, and now we try to ask for each type. We try to find the types of neurons that are both strongly downstream the PC1 DNE and the APG neurons, but also overlap with where we found the persistent activity. So we think that these are the best candidates. So we have a list now of our first candidate for carrying the persistent activity. Once we find them, then we can start working on circuits, right? So the next step is to find which exact neurons. And so I think this brings some of the strengths of Drosophila. You can really systematically use the connectome for these type of questions. Now tell me if I have 3 more minutes or that's it. Because I wanted to say something about one study that we do in the last year. And honestly, one of the motivations was some things that we saw in the connectome during the previous works, but also the fact that it takes a year to build the lab, so we have to do something that does not depend on pipettes and things like this. So we looked at the connectome. And one thing that we observed, and also other people noticed, is that if you look at the individual neurons in the fly's brain, then in many cases, not always, but in many cases, you can find neurons that have some mixed connectivity. So in many neurons, it's not the case that you have the dendrils being the inputs with postsynaptic terminals and the axons. And I know that it exists not only in flies, but it definitely exists a lot in flies. So this is one example neuron, right? This is how this neuron looks like. It's a bit hard to see, but just these are these little, dendritic trees, and this looks like the axonic area. And indeed, if you look at this area, these are indeed the outputs. So this is mapped on each synapse in one red dot. But and then you can see a lot of these inputs here. But you also see that in this area you also get inputs. So now it's becoming more complex, right? So in this case, in the area that's mostly axonic and mostly the output of the neurons, there are also inputs, and we find this quite a lot. So what we wanted to do here is to try and characterize it throughout all the fly brain and understand where does it happen and what does it mean. So we have this index that for each neuron, high index is very high polarized neuron. It's the segregation, the inputs and the outputs are very highly segregated in different compartments of the neuron. And this is the other extreme. The neurons are not polarized at all, and it's all over the place, and you can't even say where is the input and the output. This neuron, for example, is 0.5, which is quite high. Actually, 0.5 is already quite, but you can still see that there is some mixture here. So this is, and you see that this neuron that we talked about before, this one, it gets inputs from this neuron, but it gets inputs at the axon, right? At the axon. So these are, for example, between these two neurons, the connections here are axo-axonic connections. And so this is some statistics about this. This is this SI score throughout the fly brain. So you see that there are all these neurons with very low SI that are very mixed, and then these neurons are very polarized, and then there are some things in the middle. And this is looking at all the synapses of the fly brain. You can see that around 86% of them are the axonic dendritic synaptic connections, but you can see quite a few. So there are around 10% of the synapses are actually axo-axonic connections. So there is much more with it, but I will finish with just one more result, because the first question to ask is it just somehow throughout the fly brain some noise, but it's not the case. If you look at different neuropils in the fly, and here they are ordered by how much in each neuropil, right, in each area of the brain, like there is no hippocampus in the fly, but whatever is the parallel, so or not parallel, whatever the other neuropils are in the fly, so each one is a neuropil here. They are sorted by how many of the synapses there are either AA or BD, axo-axonic or dendritic. But for example, this area, this area, for example, has a lot of dendrodendritic connections, whereas this area has a lot of axoaxonic connections. It's very different between Europeans, so it's very non-random. And what we are doing now is both to try and understand, and we want to understand why is it, what's the function of this, etc. So we are trying to look at different modalities, neurons that are closer to vision, neurons that are closer to olfactory inputs, for example, and also at other motifs in the flight brain. So I'm showing it this here in case people want to tell me things about this later, because this is still halfway. But I think there is something interesting here, and these type of connectivities we can look at them systematically in the flight because we have this full connector. Yes.
Clarification. So SI score 0 here is
0 means that it's so 0 would be a case of a neuron where you have the completely mixed between where the inputs and outputs are. Like you can't even say this is the input area and this is the output area. That's the most extreme case. That's 0. Yeah. So this is. I thank the people from Princeton that are involved in the first 2 studies that I showed you, and all these people, and specifically Mara Murthy, who is now the head of the Princeton Neuroscience Institute. And this is my new lab. Very excited about this. And that's it. Thank you. Questions? Yes?
Is there any baseline difference between the locomotion of females and males?
Yeah, it's a good question. There is a small difference, but mostly in the details of the walk, and not much in the average speed. But it's a small difference. More questions?
Sorry, this is about previous work your work was based on, but as reference. So for the males, is there anything similar to what you see with the delay? That you have a specific peak at a delay?
Okay, so let's see that I understand the question. So what's similar in males is that you can also activate this small subset of neurons that drives both mating phenotypes and aggressive phenotypes. And it's persistent, and it lasts for more than 10 minutes, and it's not the same neuron because P1 is a subset, but it's from the same group of neurons. So that's the same. But if you ask if we have this phenomena in males where we have a peak that comes late, then we didn't do the work in males, but at least the work that's published, I didn't know the answer. But maybe it exists with some other perturbations.
It's not necessarily fixed to what you record or perturb.
No, so perturbing the flies, optogenetic for example, we can definitely do with freely moving flies. We don't have to open the brain to do anything. You just shine light on them. It's red shifted, so it goes through the cuticle. No problem. We can do it open loop, closed loop experiments. That's fine. But when we do imaging, we definitely put them under it, so they can't carry this miniscopes. There is some effort of subgroups to use complicated optics to follow the flies and image during social behavior, but that's very hard, and only one photon. But we don't do it.
Can you comment on the relationship between the calcium signals and electrical activity? Are these spiking neurons?
Okay, so I can't say for all the neurons in the fly, but I can say, for example, the PC2 neurons and the PC1 neurons, they are all spiking. And I know it because there are separate experiments that we did. So for the PC2 neurons and for the PC1 neurons, we also had patch clamped them, and we also found the auditory response, etc. So I know that they are spiking. But it is also true that in Drosophila there are many neurons that are not spiking. These specific ones are spiking.
And this persistent activity, for example, that you see, do you know that this is an extra spiking activity, or would it be high calcium load?
Yeah, I don't know the answer, and it's a fair question, and I think that one of the things we want to do eventually I want to do it after we know a bit more about the circuit, but I think that if we want to get to this resolution, we eventually also have to do electrophysiology, but I don't know. And it's possible. People patch clamp in flies, and that's totally doable, and I can do it, but okay, not myself, to be more honest. We can do it in the lab, we have the setup, and have to have the right person. I didn't touch that myself. More. All right.
Thank you very much.